All right, so I'm going to start with you like first question here. Okay. All right, Abdul. So my first question here is from the cell structure. It's of 2 mark question that I'm going to paste for you, and let's start to solve it. Look at this, Abdul. Here's a question. A scientist investigated the number of mitochondrial cells taken from barley plant using confocal laser scanning microscope. They recorded the number of mitochondria in each cell. They presented the data in a bar chart. Suggest why the number of mitochondria per cell increases as cell size increases. What do you think about it? Why the number of mitochondria increased as the size of these cell increased?
Is because more energy is required for
Hmm.
the biological processes
Hmm.
or like in the cell.
Like in what sense? Can you give an example where you have the large cell and there we have
Respiration, respiration.
in respiration what? I mean, I just want bit elaboration how you can justify this. It's a 2 mark question. You need 2 points. So, which 2 points that you are
Okay.
going to include here as per according to the mark scheme?
Hmm,
Yeah,
I'm writing
definitely.
while I'm thinking as well.
Yes. Good. You have an idea about it. Like, definitely the larger cells have the greater volume and therefore require more energy. Yes, this is what we already know, right? Because that has to carry out the metabolic process. Actually, this is linked to the metabolic process. So,
Yeah.
if the size is greater, so definitely that needs more energy um, to carry out the metabolic process. So one point that you need to add, that's of the greater volume, and the other point definitely that you need to add because mitochondria are the site of aerobic respiration because we have to link its function, the function of the mitochondria, and the function of mitochondria is just we know that it's a site of aerobic respiration, right? More energy is required. I think you should first you should talk about the size. Then you can go for the energy requirement during the aerobic respiration because you have to include 2 points here. So it's better as per mark scheme. Like first we should talk about the larger volume and then we can talk about the aerobic respiration, right, Abdul? As the volume of the cell increases, more energy is required through aerobic respiration for biological process in the cell, such as metabolism. Okay,
Mhm.
all right. As the volume of the cell increases, more energy is required through aerobic respiration for the biological process in the cell, such as metabolism. Yeah, it's fine. Uh, all right. Done? Or you want to add something else?
Yeah, done.
Okay. So, mitochondria are the site of aerobic respiration. So, more mitochondria needed to meet this higher energy demand, right? If you have the larger
Hmm.
size of the cell or the volume of the cells, definitely that has to carry out the metabolic process. More energy is required to carry out the metabolic process, right? So this is how you
Yeah.
can include this answer. All right, the very next question that I should add here for you is to suppose that's also a linked question. The scientists then used a flow cytometry to investigate the DNA content of the cells of the barley plant, predict how DNA content might change as cell size increases. All right. So that's also one more question.
Have you pasted the
Uh
question?
yes, Abdul, just a moment, please. Question is there for you. Take it. It's for you.
Mm.
The scientists then used flow
So the
cytometry to
science,
investigate. Yeah, you can read out, please.
yeah, the scientists then use the flow cytometry to investigate the DNA content of the cell in
Hmm.
the barley. Predict how the DNA content might change
Hmm.
as the cell size increases.
Yeah. Predict how DNA content might change as cell size increases. It's again 2 mark question. As per according to the mark scheme, you need to add 2 points here. But you should understand like what is more important that you have to start with.
Like how why the DNA content is going to be changed, like
No, that's the point. Like we need to understand why the content.
mm,
Uh, yeah. The scientists then use flow cytometry to investigate DNA content of the cells of the barley plant, predict how DNA content might change as the cell size increases. You should have an, this should be in your point. And we know when the DNA content increase in any of the cell. Yeah.
when the cell is doing the cell replication or
Yes, definitely.
mitosis. But through
Yes.
mitosis, the DNA content is not going to be changed. How the DNA content is going to be changed?
As cells grow larger, they may undergo DNA replication without cell division. Without cell division. Sir, please, you can. Sorry. Are you getting Abdul?
So the cells, as the cell grow larger, they undergo DNA
As as
replication without.
as as cells grow larger, they may undergo DNA replication without cell division, right? This is what we have, andro duplication or polyploidy. That's a part of it. So DNA content would increase. Are you getting?
Yeah, every time.
No, I think you must have an idea about it as well. What I'm talking about.
Is
You
there
know,
is it a specific experiment?
no, this is not the experiment. What I'm saying, like this one, as the cell grow larger, they undergo the DNA replication without cell division, and that's in case of polyploidy. I have told you already. Because we have taken here the example of the barley plant. But yes, one thing more, DNA content would increase with increasing cell size to support, a higher level of transcription, protein synthesis. Definitely, if the cell is larger in size, it means like that needs more proteins there. So if more proteins are required there, so definitely that has to be more, you would say, uh, replicated, and that has to go through the process of transcription. So one point that you should say, as cells grow larger, they may undergo DNA replication without cell division. We have an example of polyploidy. So for this purpose, you need to clear an idea. Polyploidy is something. Do you have an idea what is polyploidy?
I think it's not an. I never studied it. I never heard this name.
Okay, it's fine if you don't know about this, but yes, replication and I would say transcription. This is what you have already studied. So
Yeah,
keep in mind, whenever the cell size is increased, so that needs more proteins and more, different activities which are going in that cell. So that needs more transcription there. So in if the
yeah,
transcription is required more there, so it means like the DNA content, overall content is getting increased, right? So on the
yeah,
on that basis, we
yeah,
can say like that's why it may increase or it may undergo the DNA replication without cell division. Getting?
yeah. But
Ah,
if the question said the DNA content might change, so that's why I was a bit confused. That are they referring to
might change. Yeah,
mutations or
might
like?
change. Yes, exactly. Might change. So, I mean,
They should no. I thought they should have said might increase, so I would get a better answer. So when they say might change, I thought the sequence of amino acid would change.
predict how DNA content might change as cell size increases. Content might
Yeah.
change. Yes, that's going to be changed. All right.
So that I was confused that when the content of DNA is changed, this means that something mutation is happening.
Um, Mutation is not, we should not link this to the mutation, but we would say that DNA replication without cell division. This is one point that we can add. Cell DNA content would increase with increasing cell size. Why? To support
Mm.
higher levels of transcription in protein synthesis. That's what we can answer.
Okay.
Getting?
Okay. Okay.
Yeah. Um, hmm.
Yeah. Done.
So DNA content increases the cell size as cell size increases because larger cells undergo extra DNA replication to provide more copies of genes for protein synthesis, right? So this is how you can answer this question.
Yeah.
So as cell grow larger, they undergo DNA replication without cell division. The DNA content will increase to support the process of transcription in protein synthesis. Yes, that's fine. So this is how we can answer this question, right? So then after we have the next
Yeah.
question is the rise the mitochondria were once bacteria were played? It's a 4 mark question that I'm going to paste for you. That's also a linked question. Also, it's a third question that is a part of it. Look at this, Abdul. Here, read out for me, and let's try to understand what is the demand of the
So it,
question.
it is theorized. Yeah. It is theorized that mitochondria were once bacteria which were engulfed by the other prokaryotic cells. The deserting cell were the first prokaryotic cell. This is called the endosymbiotic theory. So, okay. This is, yeah. So,
Read completely
so
first. Read
basically,
out first.
yeah,
Uh,
yeah.
what's
Yeah,
next?
discuss how the structure of the chloroplast provides evidence in favor of the endosymbiotic theory of chloroplast origin. First, we can they mean that how the chloroplast was a prokaryote which was engulfed by a eukaryote. This
Hmm.
is what
Discuss how the
endosymbiotic theory
structure of
says.
chloroplast provides evidence in favor of the endosymbiotic theory of chloroplast origin. Yes. The question is,
Uh,
what is the question?
Number one, that they have how the structure of chloroplast proves that the endosymbiotic
Yes.
theory is correct, like
Okay.
that it was a prokaryote
So it means like we need to find out something in the chloroplast. What is
structure. Yeah,
the structure or the important content there, which actually proves that it was first something like a prokaryote or some and it is explained
yeah.
by the endosymbiotic theory. Hmm. So what do you think?
Number
What's
one, like if I say verbal, number one is the double membrane structure. Like chloroplast has a double membrane,
hmm, double membrane? So
and
how it justifies like it was yeah, or it is explained by the endosymbiotic theory. Hmm.
one membrane is the basically the membrane which was which was made when it was prokaryote, and
Hmm.
was membrane structure was, provided by the uh, eukaryote where it was engulfed.
Oh, okay, it's fine. One thing is this, and next one.
And the chloroplast also has its own DNA.
That's the point. That's even more important than the membrane and all that, right? Because
Yeah,
if any subcellular structure having a separate DNA, genetic material, and some proteins are there, so it means
yeah,
like that structure was somewhere uh, was surviving as an independent organism. So this is actually the proof
yeah,
for us, right?
yes. Yeah. So
Yeah.
the structure of chloroplast.
Yes. Hmm. Yes. There are 4 points that you need to add here. Okay. As per according to the mark scheme. The structure
Yeah.
of the chloroplast, which proves the endosymbiotic theory, is firstly is double membrane structure. Yeah, it's circular DNA. Hmm. Great. We have one membrane is from prokaryotic state. Hmm. Good. Membrane from is prokaryotic state. No need to explain like that's prokaryotic, but the point is we need to point out those things like membrane, DNA, circular DNA. It's independent replication. I would say division just by binary fission. That's also a part of it. I don't know it is mentioned, but we can if we have another point. So that's fine. Otherwise, it's also that they can replicate independently by binary fission. What about ribosomes here? They have the 70s ribosomes, such as bacteria for protein synthesis, because you know um,
Yeah. Yeah.
this chloroplast can
The
even
eukaryote
sense
basically have ATS.
ATS in what? In eukaryotes.
ATS in eukaryotes, but the chloroplast has 70S, so this proves that
Yes,
it.
exactly. That proves that some way it was a bacteria or a bacterial cell. Hmm. Good. Great. Now it's fine, but I have observed, Abdul, that you have started to answer structure like structured answer is quite better. Like it's easy for the examiner to understand and to read out. But Abdullah has to work on this.
Like,
Yeah. Done. I think it's fine,
oh,
right?
so,
Uh,
no, I think I have only read 2 points yet. So I'm also writing about the 70S ribosomes as well.
okay, yeah.
Like, what is the 4 point? Like, I know 3 points, but what is the fourth one?
Here I think
Like, I know
what I
about this.
can chloroplasts have their own circular DNA like prokaryotes, right?
Yeah, I have written this.
And they
Yeah.
have 70s ribosomes, same as bacteria for protein synthesis. Double membrane consistent with they have the double membrane that's consistent with engulfing by
Yeah.
endocytosis, and they can replicate
Mm.
independently. So these are 4 points.
Yeah,
Uh,
I have written this
circular
as well.
DNA
Like, chloroplast
70s.
has its own DNA, which is.
Hmm. Circular DNA, 70s ribosomes, double membrane, replicate independently. These are the 4 points that we need to add. Right, done?
Yeah, done.
Okay, that's great.
You can read if there's any problem. You can
The
say tell
structure of
me.
the chloroplast which proves the endosymbiotic theory is firstly its double membrane structure, where one membrane is from its prokaryotic state. It's a bit confusing. What do you mean by this? Where one membrane is from is prokaryotic state. Double membrane structure where one membrane
It's
is from in
from its
prokaryotic. Okay, and the
so what
other is provided by the eukaryotic
it's
host cell.
prokaryotic.
Fine, fine, got it. Because you have bit the remaining part is there. So secondly, the chloroplast has its own DNA which is divided independently from the host cell. Furthermore, chloroplasts have 70s ribosomes similar to 80s ribosomes, or we should say like 70s
So
ribosome
sorry, sorry.
similar to the
So
bacterial cell,
similar to the yeah, yeah, yeah.
similar to
I was thinking some other thing. I wrote some other thing.
mhm, such as bacteria for protein synthesis.
Done.
Okay, that's good. Now we should go for some other questions like hmm, the question is here like we have 6 mark question here. Compare the contrast the different types of fibers which make up the cytoskeleton. Okay, just a moment. One more question that's of 6 mark. And in 6 mark question, what we need to do is to explain the cytoskeleton structure. Look at this. It's a question. I think half time is past, and we just need to because we have to practice on other chapter as well, the second topic. Okay, compare and contrast the different types of fibers which make up the cytoskeleton.
Like I didn't
Figure
understand
4.1
this
shows a
question.
diagram of the structure of two centrioles. Centrioles are comprised of mitochondria. You can see here the centriole, microtubule.
Microtubules.
Yes, microtubules. Triplet is there. Centriole. Compare and contrast the different types of fibers which make up the cytoskeleton.
We didn't study the different type of fibers.
You didn't study about this?
Yeah.
What's about the centriole? Like, sorry, I
Yeah,
mean,
we know
the
about.
overall microtubules.
We only said that like microtubules, like they put like spindle fibers,
Hmm.
which are joined with the centromeres, which pull up the chromosome. That's it. Not the other types of fibers.
That's very common, like the microtubules, microfilaments, intermediate filaments. You didn't talk about this all.
No, no, we didn't study it, bro.
Right, so where's the topic? I have to get back to the specification now. Again, if this topic is included, so just a moment. Educus component one. Hmm. Cell structure and organization. Oh, sorry, it should be the very start. Hmm.
That's why I hate this exam board because this no one knows about it. Like
Hmm.
it's no very unpopular.
No, no, actually, the point is somewhere it varies. Like some, if you have studied about the overall, if you have studied about the microtubules, so definitely, we know there are different types of because if you have studied about the cytoskeleton itself, so it's not possible like somebody's if you I don't know why your instructor didn't talk about this, like what are the microfilaments or the intermediate filaments, uh, because somewhere we'll see like these are also present in the cell.
Maybe it would be a part of second year.
No, not in the
Maybe it will be a part of
in
second year
component.
content.
I mean, in the second year, the content in what sense? Because second time, because if the cell organization is given in the first in component one, so how it would be then second year second? I mean, I'm not getting your point.
Like I can tell you what they teach us in cellular structure. And they only teach us about nuclear envelope,
But it's
chromatin,
okay.
nucleolus, rough and smooth
I'm just giving
endoplasmic
you one few
reticulum.
things. Once the structure, this is what I want to tell you is if you have studied about the subcellular structures in this chapter, so cytoskeleton is also the subcellular structure which is present there. And I mean, I'm just giving you an idea about this. For example, the microtubules, as we know, they're made up of tubulin protein, right? It's a hollow tube that's of about, I would say, 25 nanometers in diameter, right? So it provides tracks
Yeah.
for organelle movement. It is involved there in the organelles movement in the cell, like vesicles, spindle fibers. So these microtubules are also involved in cell division as well, like in the form of spindle and the centrioles. This is from the
Yeah,
microtubules.
we only study microtubule in cell division. That's it. And
Uh, yes,
like my
that's its
textbook is
function.
open right in front of me.
Hmm.
Yeah, my textbook is open right in front of me. There's only microtubule in just in the cell division chapter, not over here.
Uh,
Only they talk about microtubule little bit. They talk about little bit microtubule in when they mention the centrioles.
when they talk about the centrioles, right? There's
They
not
only
any,
talk about centrioles in 5 lines. Yeah,
and it's not mentioned. Microtubules are the cytoskeleton separately there.
no,
Okay. So what's about microfilaments? It's made up of actin protein. Let me just give you an idea about it. It's thin as compared to the microtubule. So it's important in the cell shape and the movement. Like filaments are involved for the cell shape and the movement of this any sort of movements, for example, pseudopodia or the endocytosis or such things like that. So it provides support under cell membrane, right? So, intermediate filaments made up of various fibrous proteins, intermediate thickness. I think, yes, Abdul, one thing more. Do you have this book in your soft copy or something? If you can provide me.
Mm, no. It's only on the like I have a hard copy of this. I will try to find,
Mhm.
but it's very. I tried to find already, but I couldn't find online.
Yes, I tried a lot here as well from my side to find it out. Otherwise, most of the books I have from OCR, EQA, Edexcel. So, but Educus I'm not able to find, especially the cover that you have sent me. I tried my best to download this from any of the sources, but I couldn't. Uh, Somewhere it is available in, I think, Amazon or something, where in any other website. So, but I couldn't find it soft copy. Um, the cover that you have provided me already, right? So, but it's okay. If you
Yeah.
can find somewhere like any of the sources of might be your friend may have, or anybody in your instructor may also have. It's possible like your instructor should have this book with you, who is or any of.
She also uses the hard copy
But you should ask her
because they,
to provide you somewhere, like if she has in she or he has in soft form.
yeah.
Might be,
Okay.
or any more relevant to that. Not only it's not necessary like you have the same book that you have in hard form. Somewhere if you have any other
La-
that should be specific to Educus, that's my priority. Like that book should be the
Yeah, yeah, yeah.
specific to that. So any other author, like there are many other authors that they're writing. They have different even compilation of the books, especially for OCR or Edexcel especially, I would say. Anyways, if you're not much aware about this, so at least you should know about the microtubules here, right? So compare and contrast means that's just the composition and its function. These two
Yeah, yeah,
important things that we need to understand. Like somewhere you will see like a tubulin, somewhere we can see that it's made up of actin proteins. So and their function is different in the cell body, cell overall cell structure. So that's fine. Explain the importance of centriole in animal cells. This is what we can talk about. It's a FOMA question, so I hope this is what you already know about it as far as centriole is concerned. And keep in mind,
yeah,
Abdul, books are
yeah, yeah,
just the compilation of, for instance, like if I am teaching educus from long times, like then I can say one thing like I can compile book or to content overall, right? So
yeah, yeah.
that's not the 100% like this is the book and this book will be followed by any of the examiner. This you might that's the general
Yeah,
thing like that can give you an idea.
yeah, yeah.
Okay, this book is bit relevant to my content, but you should not. You should. I would not say so.
Rely.
100% you can't rely on this. Somewhere you need to go to check your. I mean,
Yeah, I read my specification as well. Yeah.
yes,
To
specification is important. That's. Yes, I just want to go there on the specification. What's in that? Like, I just want to get into that. Do you have any, like, cell structure and organization, working scientifically, mathematical scale, how science works? Learners should be able to demonstrate and apply their knowledge and understanding of the structure and function of the fall mitochondria, endoplasmic reticulum, ribosomes, Golgi operators, lysosomes, centrioles, chloroplast, vacuoles, nucleus, chromatin, nuclear envelope, nucleolus. Oh yeah, it's fine. Uh, the structure of the prokaryotic cells in viruses, cell theory, and the similarities and differences in the cell structure of the eukaryotes and prokaryotes and and of viruses, including the examination of the range of electron. Oh yeah, I think a cytoskeleton is not mentioned here. Abdul,
yeah,
then I don't think so. Like we
yes.
need to go for such things. Mm, light microscope, low. Okay, that's fine. That's specified for practical work. Cell membrane and transport. That's the second part of it. Okay, then there's no need to go for to have this question. Yeah, that's fine. Because ribosomes, centriole is there or not? Where is centriole? Chromatin, nuclear
Central is
envelope,
there.
centriole is present here. Where is that? Ribosomes, Golgi operators, yeah, centriole is here. Yes, as far as centriole is concerned, all right, we can talk about the microtubules there. That's fine. Okay, so next question is that's explain the importance of centrioles in animal cell. Let me paste this for you. Yeah, Abdul, I think that's even we should not be confused because specification is in front of us. Like that's fine. If this question is not in detail from your side, so it's fine. Look at this. Explain the importance of centrioles in animal cell. It's a 4 mark question. It means like you need to add 4 points here. Explain the importance of centriole in animal cells. So
Oh,
what do you
you
what
know,
do you
meanwhile, I'm solving the question. Like there's one website provided by my college. Like it's a Cube, QUBE. It's like it's specific to my college, and I can give you my credentials, and you can log in, and I will tell you where
Yes.
you have to go. Then you will find a book related to educast biology.
Oh, great. So it's fine. Like if you
So
have their online, it's available online. Their book
yeah,
as
yeah,
a complete book. Okay, that's fine. Yeah, that's what I would say. Like if you have the credentials with you, you should share with me. So we will be more specific to that. And yes, the second important thing
yeah,
again, this is your Bible. Specification is your Bible. I mean, we should not, even your examiner is not able to go out from here because he's bound to
yeah,
make a paper from here. So yes, we can
yeah,
consult any of the book, but again, we need to we should always keep this next time in our upcoming classes, I'll try my best to keep open this specification in front of me, so there should not be any question that is going out, or we should not miss any specific topic from here. And yes, you should provide me. I mean, you should send me in my chat, Zoom chat, credentials if you if available to you right now.
Yeah. Yeah, I can.
Okay, send it to me. Then after we'll go for it. Oh, good. All right. Let me. Oh, great.
The second one is the password.
Yeah. Okay. Fine. Let me open this. Yeah. Just cube or something like any other. Because once I'm putting cube here, so many of the things that's coming in front of me. Is this not possible for
When
you
you
to
say
send me a
cube,
link?
hard lock wale.
Just send me a link as well.
Oh yeah, yeah, I can do that.
Cube agentic analysis platform, no code, maths world.
Like cube is a okay. Like 1 second. I'm sending you a link of a website. You can log in over there.
Yeah.
It's you ebook center. You can just open this.
Okay, okay, fine. Copy this link. I'm getting back to my Google point. Okay, that's fine. All right, Harlow. Yes, it's gone. Wait a second. All right, now I can do it. Mm. Okay, next. Password. Copy. Sign in. No, it's what your account password is incorrect. In what sense it's incorrect? Why it is incorrect?
Let me check.
Can you please just check your
One
password? I
second.
think it is incorrect here. No, it's not accepting. Your account or password is incorrect if you don't remember your password, so you can change it as well. Or reset it now. You have an option there. Harlow College. Great.
Uh, Write in password you can write 110508 or 1105 2008. I don't know why it's not. I will check. Or you can try 110508.
110508.
Yeah,
110508.
8.
No, it's not accepting. Oh, your account or password is incorrect. Again, the same. You can change it.
Or you
It's
can
it
write.
does. Why so like
No,
you
the
should.
problem is if I change it from there, like the password is linked from all of my credentials. Like once I change this, like I have to do a lot of like changing my Wi-Fi password, which is provided by college, and so many things.
Oh my
Uh,
goodness. Okay.
or
So why you not
or you can write try
mhm.
or you can try 1105 2008. Just
11
the last time. Otherwise, then
11
05 2008.
11 2008. I can't see it here. No, it's same. Why not you? I think you should try
Okay,
on
then
your side. I think you should try on your side. So if it's working, it's fine. Otherwise, you can confirm it from your side and then you can share with me. Try once on your side if it's not getting open. So then after you can confirm it and then share it with me. That's fine. Otherwise, it's because I've tried here, nothing is work. It's okay. One more.
Okay, we can do on the next class. We will do on the next class, or when once I
Okay.
get it, I will
O-
convey that.
okay, that's fine. Otherwise, let's try to complete these two questions. Then after I'll send you on today rest of the questions in the form of worksheet just by consulting your the specification, right? So you can download that from the portal and to solve it from your side, and you can upload it again as well. Okay, all right. So I'll send you
Uh,
that worksheet as well. I mean the remaining questions from there. Otherwise, just try to solve this question first. Explain the importance of centriole in
yeah.
animal cells. There are four mark questions, so you need to add 4 points here.
Like I said, centrioles are two ring of microtubules. During cell division, centrioles organize the microtubule that make up the spindle
Mm.
to to pull the sister
Mm-hmm.
chromatids to each pole of the cell. Yeah, done.
So what's the first point? Centriole organization are organized microtubules during cell division. This is its first role, right? And the second role is they
Yeah.
form a spindle fibers that attach to the chromosomes and ensure the correct separation to daughter cells, right? And one more thing, centrioles also act as basal bodies to produce cilia and flagella. You know, if you remember these structures, they also have the centrioles in its base, so which are important for the cell movement and fluid transport. So I think 3 points.
No, but flagella is, I think it's in prokaryote. I think that in animal cell where we don't need flagella, isn't
But
it?
yes, we are talking about the animal cells. In case of animal
Yeah.
cells, we just need to talk about the centrioles organization, the microtubules during the cell division, right? Uh,
Yeah.
is this not in case of cilia? Like cilia are also present in animals as well. If you remember, like in the cilia are present in the small intestine, where their function is to increase the surface area for the absorption of the different content which is present in the small intestine. So cilia is there, right?
But what is the relation between cilia and centrioles?
Relationship is that the basement body, the basal body of the cilia is made up of centrioles. That is, centrioles also act as a basal bodies. Just if you can look into the structure of the cilia, so you will see like just at the base of that outgrowth, that cilium, uh, the structure which is present there, that is centriole. So that's why we would say like
Uh,
centrioles also act as a basal bodies to produce cilia, and
uh,
which is important for the cell movement and the fluid transport. This is what's its function. All right. So
yeah,
spindle operators, spindle fibers attachment to the chromosomes, and centrioles also act as a basal bodies. So these three points that we can include for the animal cell, right? Okay,
yes.
so the next question here is like we have, suppose, eukaryotic cell contains many membrane-bound cellular components. Discuss with examples the importance of membrane-bound cellular components for the cell function. Suggest why prokaryotic cells do not have membrane-bound organelles. Have you studied about this? Eukaryotic cell.
Yeah, but
Hmm.
I
Let,
know
let,
that they don't have membrane-bound organelles.
hmm. Yes, but oh, sorry, what is this? Okay, just a moment. Here it is. Okay, just try to solve it. It's a 6 mark question. It will take time, but we'll try to complete this. So just write the points, but you should understand. Then you can.
Eukaryotic cells contain many membrane-bound or cellular components. Discuss with example the importance of membrane-bound cellular components for cell
For cell
function.
function. Um.
Yeah.
Okay. As per mark scheme, we should check it.
The memory
Hmm.
uh,
It's quite.
organelle.
Hmm. Compartmentalization. Membrane-bound organs allow compartmentalization. Yeah. Discuss with examples the importance of membrane-bound cellular components for cell function. I mean, you know what? You need to add, suppose, nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, lysosomes, like all of these structures, you need to take one by one and to give some bit a short explanation on that. For example, the mitochondria have double membranes. The inner membrane provides large surface area for ATP production during aerobic respiration. I mean, we just need to take one example from there and to talk about its specific function there. So this is how we can answer this question. This is a very straightforward question, not like where you need some trick. So just uh, we just name few of the organelles from there, like nucleus, mitochondria, chloroplast, endoplasmic reticulum, Golgi apparatus, lysosomes as well. Like these 5 uh, of the organelles that we need to talk about, right? So this is what you can do after that. One more important thing is suggest why prokaryotic cell do not don't have membrane bound organelles. Yeah, what do you think about it?
Because they have a less surface area.
Less surface area in what sense? Less
Because
surface
they are
area. How you? Hmm?
they are they are very small in size.
Yes, in specification, you have the difference in the cell structure of the eukaryotes and the prokaryotes. That's
Yeah.
you can
The
talk
cells
about.
are very small.
Yes, that's fine. This is what we can add. Like uh, prokaryotic cell is quite simple. Compartmentalization required where you have to do a lot of functions. Compartmentalization itself is saying like every part of it has to perform a different function. Like if you can see the eukaryotic cell, that's quite complex and it's performing number of functions there, right? So mitochondria had
Yeah.
its own function, endoplasmic reticulum has its own function, and the overall structure of the cell is quite complex. However, in case of prokaryotic cell, we don't need too much complexity there because it's already very simple cell. It's not involved in much of the functions. So we would say like the prokaryotic cell are much smaller, are simpler. So compartmentalization is not necessary there, or it's not important there, right? And you can add like one more point for this, as per according to the mark scheme. It's of 2 mark question. Yeah, for 2 marks, you need
What
to
about the
add.
6 marker one? We haven't wrote any. We should start the 6 mark. I've done the 2 marker one. I wrote the cells are very small inside and have a very simple structure, so the
Yes,
compartmentalization are not
yes.
essential.
In case of 6 mark, I have told you already, like you need to take different organelles and talk about their function. Like take nucleus from there, that's surrounded by double membrane. You should say like which protects DNA and controls gene expression. Like we need to talk about why nucleus is present. why chloroplast has its structure, membrane bound structure. You know very well, like in chloroplast, if you can say, like, have membranes that house photosynthetic pigments, like enabling photosynthesis. Endoplasmic reticulum membrane, we know the rough endoplasmic reticulum are synthesis of proteins which are involved there. So every membrane has some specific function in the cell, right? In case of mitochondria, if we can recall that concept, so inner membrane provides a large surface area for ATP
Yeah, you done.
production, right?
Be quick. My time.
Yes.
Okay. I think Abdullah is there. All right. So then Abdul, I think what you need to do is take some
I will.
organelles name and write their functions. This is
Okay.
this is how you can
1 minute
you
pass,
can
bro.
fulfill the requirement
Be
of
quick.
this question. It's a 6 mark question, right? So
Okay.
yeah, it's done from your side. I think now it's Abdullah's turn. So he's waiting. I
So
I
I
can
will
I
do.
can
I
yeah.
I will do. I will do it after the. But I
And
have
please,
a maths right now, so
and Abdul,
I will do after math.
okay, okay, Abdul, and please keep an eye on your portal as well. So I will share rest of the questions there, so you can download from that portal and to attempt from your side. Okay,
Yeah.
okay, all right.
Can you
So
please move to? Can you please move me to Miss Amber's room? Like, can you do it from your side?
Miss Amber's room, I can't do anything from my side. I think this is what uh I think the
Okay.
admin is doing. All right. Okay, take care of
Okay.
yourself,
Okay.
Abdul. See you tomorrow. I mean, see you next
Allah
week.
Hafiz.
Okay, take care. Allah Hafiz.
Allah Hafiz.